Welcome everyone. Today we're diving into Module 3, Enzymology. I'm excited to guide you through this fascinating topic. This is Study Session 1, where we'll explore the chemistry of amino acids and proteins, focusing on the classification of lipids. This is part of BCHM 241, the biochemistry of macromolecules. So, let's get started and discover how these essential biomolecules function and interact. Let's take a closer look at the classification of lipids. Lipids can be broadly divided into three main categories: simple, complex, and derived. Simple lipids mainly consist of fats and oils, which are primarily triglycerides. Complex lipids include molecules like phospholipids and glycolipids, which have additional functional groups. Derived lipids are substances that are chemically modified from the simple and complex lipids, such as steroids and fatty acids. Speaking of fatty acids, these are fundamental building blocks of many lipids. They can be saturated, meaning they have no double bonds, or unsaturated, which contain one or more double bonds. This distinction affects their physical properties and biological functions significantly. Eicosanoids are another important class, derived from fatty acids, and they include prostanoids and linear eicosanoids. Prostanoids, for example, are involved in inflammation and other signaling processes. Linear eicosanoids are also derived from fatty acids and participate in various physiological responses. Moving on to triacylglycerols, these are the main storage form of fats in the body. They consist of three fatty acids attached to a glycerol backbone, serving as a dense energy reserve. We also have steroids, which include molecules like bile acids and steroid hormones. These are vital for digestion, hormone regulation, and cell membrane structure. Finally, lipoproteins such as HDL, LDL, VLDL, and chylomicrons are essential for lipid transport in the bloodstream. Each type has a specific role. For example, HDL is known as good cholesterol, while LDL is often called bad cholesterol because of its association with cardiovascular disease. Understanding these classifications helps us grasp how lipids function in health and disease, tying into the broader themes of metabolism and physiology. Lipids are a diverse group of molecules that are relatively insoluble in water, and they're closely related to fatty acids. You'll notice that they dissolve well in nonpolar solvents like benzene, ether, chloroform, and acetone, which highlights their nonpolar nature. Unlike proteins or nucleic acids, lipids are not polymers. You might wonder why that is. Well, even though they are large and complex molecules, they aren't built from repeating units or monomers. Instead, they are considered macromolecules because of their size and complexity, but they don't have the repetitive structure characteristic of polymers. This distinction is important because it influences how lipids function and how they are constructed in biological systems. So, understanding that lipids are heterogeneous, nonpolar, and nonpolymeric helps us appreciate their unique roles in cell membranes, energy storage, and signaling. Let's take a closer look at the classification of lipids. Lipids are a diverse group of molecules, and understanding how they are categorized helps us better grasp their functions in the body. Broadly, lipids can be grouped into several main categories. First, we have fatty acids, which are the building blocks for many other lipids. Then, there are triglycerides, also known as fats and oils, which serve as energy storage molecules. Next, phospholipids are essential components of cell membranes, providing structure and fluidity. Steroids, another major class, include cholesterol and hormone precursors that play key roles in signaling and regulation. Lastly, we have other complex lipids, which may include various specialized molecules with unique functions. Recognizing these classifications not only helps us understand their individual roles, but also how they work together to support our health and metabolic processes. This classification underscores the importance of lipids in biological systems and sets the stage for exploring their specific functions in more detail. So, based on the type of alcohols involved, these lipids are further subclassified. Essentially, they are esters of fatty acids with various alcohols. This means that fatty acids combine with different alcohol molecules to form these esters. When we talk about simple lipids, we're referring to these basic structures that are foundational in lipid chemistry. Understanding this classification helps us see how diverse lipids can be, yet how they all share this common ester bond between fatty acids and alcohols. This concept ties into the overall theme of how lipids are built and how their structure influences their function in biological systems. Let's take a closer look at fats and oils. Fat, as you might know, is a type of triglyceride that is solid at room temperature, whereas oil is also a triglyceride but remains liquid under the same conditions. The key difference lies in their chemical structure and how the fatty acids are arranged. Both fats and oils are esters formed from fatty acids and glycerol, which is a type of alcohol. These are known as neutral fats or triglycerides, and they serve as important energy stores in our bodies. Understanding these distinctions helps us grasp how fats function in nutrition and biochemistry, connecting to the bigger picture of how our bodies utilize different types of lipids. Let's take a closer look at some key examples of waxes. First, we have lanolin, which is derived from wool grease and is widely used in cosmetics and skincare products. Then there's beeswax, a natural wax produced by honeybees, known for its protective and emollient properties. We also have whale sperm oil, which has historically been used in various applications, although its use is now more limited due to conservation concerns. Another important category includes esters of fatty acids with higher molecular weight monohydric alcohols. These esters are often valued for their stability and unique physical properties. Overall, these examples illustrate the diversity of waxes, natural and synthetic, that serve various functions across industries. Understanding these different types helps us grasp how waxes are formulated and utilized, fitting into the broader picture of materials with specific physical and chemical characteristics we rely on. Now, let's take a closer look at complex lipids. These are classified based on the type of prosthetic group they contain. Essentially, they are esters formed from fatty acids and alcohols that have additional prosthetic groups attached. These prosthetic groups are what give complex lipids their unique functions and characteristics. Understanding this classification helps us see how diverse and specialized these lipids are in biological systems. Remember, the key point here is that the nature of the prosthetic group determines the specific role and properties of each complex lipid, tying back to the overall theme of lipid diversity and function in the body. Now, moving on to slide 9, let's talk about how phospholipids can be classified based on the type of alcohol present. Essentially, phospholipids are composed of three main components: fatty acids, an alcohol, and phosphoric acid. The key point here is that the alcohol component varies, and this variation is what allows us to classify different types of phospholipids. When we look at the structure, you'll notice that the fatty acids are attached to the alcohol, which in turn is linked to phosphoric acid. The specific type of alcohol, whether it's serine, choline, ethanolamine, or inositol, determines the class of phospholipid. This classification is important because it influences the properties and functions of these molecules, especially in cell membranes. Different alcohol groups can affect membrane fluidity, signaling, and interactions with other molecules. So, in the context of the overall theme, understanding this classification helps us appreciate how diverse phospholipids are and how their structure relates to their role in maintaining cell integrity and facilitating communication within the body. Let's take a closer look at sphingomyelins, which are a type of sphingolipid. They play a crucial role in cell membrane structure and signaling. Moving on, we see phosphatidylcholine, a major component of cell membranes, known for its role in membrane integrity and lipid metabolism. Next is phosphatidylethanolamine, which also contributes to membrane fluidity and is involved in various cellular processes. Phosphatidylserine is particularly interesting because it flips to the outer membrane during apoptosis, signaling cell death. Then there's phosphatidylinositol, which is vital for cell signaling pathways and membrane dynamics. We also have plasmalogens, a special class of phospholipids with antioxidant properties that protect cells from oxidative stress. Cardiolipins are unique lipids found mainly in the inner mitochondrial membrane and are essential for mitochondrial function and energy production. Now, a quick note about the alcohol components. In sphingolipids, the alcohol is sphingosine, whereas in glycerophospholipids, the alcohol is glycerol. Let's differentiate the two main categories here. B. Sphingophospholipids, which contain sphingosine as the alcohol backbone, and A. Glycerophospholipids, which are built on glycerol. Understanding these differences is key to grasping the diversity and function of cell membrane lipids, which ties into the overall theme of membrane structure and function in cell biology. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at glycolipids. An important example of these molecules are cerebrosides and gangliosides. What sets glycolipids apart is that they do not contain a phosphate group, unlike phospholipids. Instead, their structure includes fatty acids combined with alcohol, specifically sphingosine, which is a key component. Attached to this backbone is a carbohydrate group that contains a nitrogen base. These structural features are essential because they influence how glycolipids function, especially in cell membranes and signaling. Understanding these components helps us see how glycolipids contribute to cell recognition and communication, fitting into the bigger picture of lipid diversity and function we've been exploring. Let's talk about lipoproteins, which are essential for transporting lipids through the bloodstream. As you can see on this slide, they are composed of a lipid core with a prosthetic protein group, which helps them interact with cell receptors. This structure allows them to carry fats efficiently to various tissues. We start with chylomicrons, which are formed in the intestines after a meal and primarily transport dietary fats. Then, we have VLDL, or very low-density lipoproteins, produced in the liver to carry triglycerides synthesized there to peripheral tissues. Next, LDL, or low-density lipoprotein, is often called bad cholesterol because it delivers cholesterol to cells, and excess LDL can lead to plaque buildup in arteries. Conversely, HDL, or high-density lipoprotein, is known as good cholesterol because it helps remove excess cholesterol from the bloodstream and brings it back to the liver for disposal. Understanding these different lipoproteins is key to grasping how our body manages lipid transport and how imbalances can impact cardiovascular health, which ties directly into our overall discussion on lipid metabolism and disease prevention. Now, let's talk about derived lipids. These are the products we get when we hydrolyze group 1 and group 2 lipids. Essentially, these derivatives still maintain the key characteristics of lipids, even after breaking down the original molecules. It's important to understand that through hydrolysis, we can analyze and identify the building blocks of lipids, which helps us better understand their functions and roles in biological systems. So, in essence, derived lipids give us valuable insights into the structure and behavior of lipids, connecting directly to the overall theme of lipid metabolism and function. Today, I want to talk about the functions of lipids. Lipids are incredibly important molecules in our body, and they serve a variety of key roles. First and foremost, lipids are a major source of energy. They store energy efficiently, providing more calories per gram than carbohydrates or proteins. When our body needs energy, it taps into these fat stores. Another critical function is their role in forming cell membranes. Lipids, especially phospholipids, are fundamental components of the cellular membrane, helping to maintain cell integrity and regulate what enters and exits the cell. This structural role is vital for cell function and communication. Lipids also have insulating properties. They help protect our internal organs and regulate body temperature by acting as an insulative layer. This is especially important in maintaining homeostasis. Additionally, lipids are involved in signaling processes. Steroid hormones, which are derived from lipids, act as messengers that coordinate complex processes like growth, development, and immune responses. Connecting these points to our overall theme, understanding the functions of lipids highlights their versatility and essential roles in maintaining health and supporting life processes. They are not just energy reserves, but active participants in structural and regulatory functions within our bodies. Let's take a closer look at the functions of lipids. Lipids play several crucial roles in our bodies. First and foremost, they are essential for storing energy efficiently. Unlike carbohydrates, lipids provide a dense source of energy that our bodies can tap into when needed. Secondly, lipids are vital components of cell membranes. They help maintain the structure and integrity of cells, acting as barriers that regulate what enters and exits. This is fundamental to cell function and communication. Another key point is that lipids serve as signaling molecules. Hormones like steroids are lipids that help regulate various physiological processes, including growth, metabolism, and reproduction. Lastly, lipids also have insulating and protective functions. They provide thermal insulation to keep our bodies warm and cushion vital organs against injury. Connecting this to our overall understanding, lipids are indispensable not just for energy storage, but for maintaining cellular health, facilitating communication within the body, and protecting our organs. They truly are multifunctional molecules that support our overall health and functioning. Fatty acids are naturally occurring carboxylic acids, and they typically have an unbranched carbon chain. When we talk about fatty acids, it's important to note that they can have either an even or odd number of carbon atoms. However, during their biosynthesis, they almost always contain an even number of carbons. This is because the pathway for synthesizing fatty acids usually adds 2 carbons at a time. Now, the length of these carbon chains varies quite a bit. Long-chain fatty acids, which range from about 12 to 26 carbons, are commonly found in meats and fish. These are vital components of many biological membranes and energy storage molecules. On the other hand, medium-chain fatty acids, with 6 to 10 carbons, and short-chain fatty acids, with fewer than 6 carbons, are primarily found in dairy products. These shorter chains are often easier for the body to digest and utilize quickly for energy. Finally, fatty acids can be classified into 2 main types: saturated and unsaturated. Saturated fatty acids have no double bonds between carbon atoms, making them solid at room temperature, while unsaturated fatty acids contain 1 or more double bonds, which introduce kinks in the chain and typically make them liquid at room temperature. Understanding these differences and the sources helps us grasp how fatty acids contribute to nutrition and metabolism, fitting into the broader picture of lipid biology and health. Let's talk about unsaturated fatty acids first. These are molecules that contain 1 or more double bonds in their carbon chain. Because of these double bonds, the molecules have a bend or kink, which affects how they pack together. Unsaturated fatty acids can be classified further into mono- and polyunsaturated fatty acids. An example of a monounsaturated fatty acid is oleic acid, which has 1 double bond. Polyunsaturated fatty acids, on the other hand, have multiple double bonds, making them more flexible and often more beneficial for health. Now, moving on to saturated fatty acids. These are fatty acid chains that contain only single bonds between the carbon atoms. Because there are no double bonds, these molecules are straight and can pack tightly together. An example here is palmitic acid. The saturation level of a fatty acid, meaning whether it has double bonds or not, plays a significant role in its physical properties and health effects. Tying this back to the overall theme, understanding the saturation level of fatty acids helps us grasp their different functions and impacts in the body. Unsaturated fats tend to be healthier and are often found in plant-based oils, while saturated fats are more common in animal fats. Recognizing these differences is key to making informed dietary choices and understanding how fats influence our health. Fattig syra blur ogsa classifiesert, bassert pa lengden av carbonjetin. Nar Roman 6 snacker lengden pa hydrocarbonjetin. Referer Roman 6, till H4 mange carbonatomer, somer coblet salmon I molkeilet. Den egenskapen veldig viktig 40 den paverker, H4den fetsiren opforer seg. Som for exempel deres, smeltepunkt og luselighet. Quarter kjetter har tendens, till a ring vermer flytend vid romtemperatur, mens langer kjetter ofter fast eller fast vid sam temperatur. Det en av grunning, till it ulike fetstoffer har ulike egenskaper og bruksomrater. Sa, nar Roman 6 studerer fetsura, det viktig a ring merkseg, et karbonjettens lengd spiller en nakelroll forden de opfor, seg I kroppen og I mat. Let's talk about how fatty acids are named. They are named based on the hydrocarbon chain they are derived from. For example, the names of saturated fatty acids typically end with enoic, like in stearic acid. On the other hand, unsaturated fatty acids end with enoic, such as oleic acid. It's also important to note that fatty acids often have common names that are widely used in everyday language and scientific contexts. Understanding this naming system helps us grasp their structure and function better, which ties into the overall theme of how these molecules impact our health and biology. This slide doesn't contain enough information. That's why we're moving to the next slide. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about how carbon in fatty acids is numbered. The numbering starts from the carboxyl carbon, which is the very first carbon in the chain. We can label this carbon as either one, using an Arabic numeral, or as alpha, which is a Greek letter. From there, we proceed along the chain toward the methyl end, which is the last carbon, known as the omega carbon. Sometimes, instead of numbering from the carboxyl end, we can do the reverse and start from the methyl end. In that case, the carbons are numbered as W1, W2, W3, and so on, all the way toward the carboxyl group. This alternative system is useful in certain contexts, especially when discussing the position of double bonds or other features relative to the methyl end. Understanding these different numbering methods is important because it helps us precisely identify and describe the structure of fatty acids. It connects directly to how we analyze their properties and functions in biological systems, which is a key theme of this presentation. There are two essential fatty acids that are really important for our health. When I say essential, I mean we actually need to get these from our diet because our bodies can't make them on their own. The first one is alpha-linolenic acid, or LNA, which is an omega-3 fatty acid. Omega-3s are known for their benefits in supporting brain health and reducing inflammation. The second essential fatty acid is linoleic acid, or LA. This is an omega-6 fatty acid, and you'll find it quite abundantly in foods like soy oil, sunflower seeds, pumpkin seeds, sesame seeds, corn oil, and most nuts. These fats are crucial for maintaining cell structure and function, so including them in your diet is key for overall health. Now, let's talk about the functions of essential fatty acids, or EFAs. These nutrients are absolutely vital for maintaining the structure and proper function of cell membranes. Without them, cells can't operate effectively. EFAs also play a key role in the transport of cholesterol, helping to move it through the bloodstream to where it's needed or to be excreted. This is crucial because cholesterol is involved in many processes, including hormone production and cell integrity. Another important function is their contribution to the formation of lipoproteins, which are the particles responsible for carrying fats and cholesterol through the blood. This process is essential for fat metabolism and overall cardiovascular health. EFAs also help prevent fatty liver, a condition where fat accumulates excessively in the liver, impairing its function. By supporting healthy fat metabolism, they help keep the liver functioning properly. Lastly, EFAs are necessary for synthesizing a group of compounds called eicosanoids. These are powerful signaling molecules that regulate inflammation, blood flow, and immune responses. So, in essence, EFAs are foundational not just for cell structure, but also for vital signaling pathways that keep our bodies balanced and responsive. Let's take a closer look at some of the consequences that can arise from essential fatty acid deficiency. One of the key issues is growth retardation, which means that growth in children can be significantly slowed down or stunted. This highlights just how important these fats are for normal development. Poor wound healing is another serious problem, as essential fatty acids play a vital role in the body's ability to repair tissue. Without enough of them, wounds can take longer to heal, increasing the risk of infections and complications. You might also notice skin issues like dermatitis, which is inflammation of the skin, and hair loss. These symptoms reflect the crucial role that essential fatty acids have in maintaining healthy skin and hair. In addition, deficiency can affect internal organs, leading to kidney and liver diseases, which can be quite serious if left unaddressed. Reproductive health can also suffer, resulting in infertility, which underscores how essential these fats are for hormonal balance and reproductive functions. Mental health isn't spared either. Depression can develop, indicating that essential fatty acids are important for brain function and mood regulation. Overall, these points emphasize the importance of maintaining adequate levels of essential fatty acids, as deficiencies can impact multiple systems in the body, reinforcing their role in overall health and well-being. They are esters of alcohol called glycerol and fatty acids. This means that fats in our body are essentially made up of these molecules combined together. Their primary role is to serve as the storage form of fats, and you'll find them mainly in adipose tissues, which are the body's fat reserves. One of the key points to remember is that these fats are a highly efficient energy source. In fact, 1 gram of fatty acids can release about 9 kilocalories of energy, which is roughly 2.5 times more than proteins or carbohydrates that yield about 4 kilocalories per gram. This high energy density makes fat an incredibly effective way for the body to store energy in a compact form. When the body needs energy, triacylglycerols, or TAGs, can be hydrolyzed, that is, broken down, to produce diglycerides, monoglycerides, fatty acids, and glycerol. Understanding this process helps us see how fats are mobilized and utilized during periods of fasting or increased energy demand. Overall, triacylglycerols are the body's most efficient energy reservoirs, which plays a crucial role in maintaining energy balance and supporting various bodily functions. Let's take a closer look at the structure of TAG. As you can see, understanding how it's built is key to grasping its overall function and effectiveness. The structure is designed to be both comprehensive and flexible, allowing it to adapt to various scenarios. At its core, TAG consists of several interconnected components that work together seamlessly. These components include the core framework, the supporting modules, and the integration points that connect everything. By examining each part, we can better understand how they contribute to the system's robustness and efficiency. Ultimately, this structure is what enables TAG to deliver its full potential, ensuring it remains a versatile and reliable tool within our overall strategy. Recognizing these connections helps us appreciate how each element plays a vital role in the bigger picture. TAG can exist in 2 forms, as fat or oil, depending on their composition. It's important to remember that fats and oils can develop an unpleasant smell and taste if they are stored for a long time under moist conditions. This change happens due to a process called hydrolysis and oxidation, where fatty acids are released and then further break down into compounds like aldehydes and ketones. These compounds are responsible for the off flavors and odors we associate with rancidity. Essentially, rancidity is a chemical reaction that affects the quality and safety of fats and oils, which is why proper storage is so crucial in preserving their freshness and nutritional value. Connecting this to our overall theme, understanding these chemical changes helps us better handle and store fats and oils to maintain their quality for longer periods. Lipoproteins are essentially molecular complexes made up of lipids and proteins working together. They play a crucial role as transport vehicles for lipids within our bloodstream. Think of them as the delivery trucks that carry essential lipids like cholesterol and triacylglycerol to different tissues throughout the body. Once they reach their target tissues, these lipoproteins enable the lipids to be utilized effectively, whether for energy, cell repair, or other vital functions. Understanding lipoproteins helps us see how our body maintains lipid balance and supports overall metabolic health, which ties back to the larger theme of lipid transport and regulation. Let's take a closer look at the structure of what we've discussed. The structure is essentially the framework that holds everything together. It helps us understand how the different components fit and work in harmony. By examining this, we can see how each part contributes to the bigger picture. This is crucial because a well-organized structure makes the information more accessible and easier to follow. It also highlights the logical flow, which is key to effective communication. Remember, understanding the structure reinforces the overall theme of clarity and coherence in our presentation. So, paying attention to this aspect ensures that our message is both impactful and easily understood. Today, I want to talk about the 5 main types of lipoproteins that play a crucial role in our body's lipid transport system. First, we have low-density lipoproteins, or LDL, which are often called the bad cholesterol because they can contribute to plaque buildup in our arteries. Then, there are very low-density lipoproteins, or VLDL, which primarily carry triglycerides from the liver to tissues. Next up are high-density lipoproteins, or HDL, known as the good cholesterol because they help remove excess cholesterol from the bloodstream and bring it back to the liver for disposal. We also have chylomicrons, which are responsible for transporting dietary lipids from our intestines to other parts of the body. Understanding these classes of lipoproteins is essential because they work together to maintain balanced lipid levels and support overall cardiovascular health. Recognizing the differences among them helps us better grasp how lipid transport impacts our health and how lifestyle choices can influence these processes. So, keeping these key players in mind is central to understanding the broader picture of lipid metabolism and disease prevention. Let's take a closer look at lipoproteins, starting with chylomicrons. These are classified as very low-density lipoproteins, or VLDL. They are mainly responsible for transporting dietary fats from the intestines to various tissues in the body. Because they are less dense, they contain a high proportion of triglycerides compared to proteins. Moving on, we have LDL, or low-density lipoprotein. Often called the bad cholesterol, LDL carries cholesterol from the liver to the cells. Its density is higher than VLDL because it has a relatively higher proportion of cholesterol and less triglyceride. Elevated levels of LDL are associated with an increased risk of cardiovascular disease. Finally, we have HDL, the high-density lipoprotein, often referred to as the good cholesterol. HDL plays a crucial role in transporting excess cholesterol from the tissues back to the liver for disposal. Its high density indicates a higher protein content relative to lipids, which makes it effective in reverse cholesterol transport. Understanding the composition of these lipoproteins is key because it helps us grasp how fats and cholesterol are transported and managed within our bodies. Each type has a unique role, and their balance is essential for maintaining cardiovascular health. Now, let's talk about ultracentrifugation of lipoproteins. This is a key technique used in biochemistry to separate different types of lipoproteins based on their density. Essentially, by spinning samples at extremely high speeds, we can isolate various lipoprotein classes like chylomicrons, VLDL, LDL, and HDL. This process relies on the fact that each lipoprotein has a unique density, which is influenced by their lipid and protein composition. Ultracentrifugation helps us analyze their structure and function more precisely, which is vital for understanding their roles in lipid transport and cardiovascular health. Connecting this to the overall theme, understanding how to effectively isolate and study lipoproteins gives us deeper insights into their contribution to disease processes and potential therapeutic targets. It's a foundational technique that underpins much of our research in lipid metabolism. Let's take a closer look at how lipids are transported in the blood. Starting with triacylglycerol transport, we have chylomicrons, which are responsible for carrying dietary triglycerides from the intestines to other tissues. These particles are essential for delivering the fats we consume daily. Then we have VLDL, or very low-density lipoproteins, which mainly carry triglycerides that are synthesized endogenously in the liver. These lipoproteins help distribute the body's internal energy stores. Moving on to cholesterol transport, LDL, or low-density lipoprotein, primarily carries free cholesterol. This form of cholesterol is crucial for cell membrane structure and hormone synthesis, but high levels of LDL can contribute to atherosclerosis. In contrast, HDL, or high-density lipoprotein, primarily transports esterified cholesterol. HDL is often called the good cholesterol because it helps remove excess cholesterol from tissues and arteries, transporting it back to the liver for excretion or recycling. Understanding these different lipoproteins and their roles is fundamental for grasping how our body manages fats and cholesterol, which is central to overall cardiovascular health. Today, I want to focus on cardiovascular disorders, which are a significant part of overall health concerns. These disorders include a range of conditions that affect the heart and blood vessels, and they are among the leading causes of morbidity and mortality worldwide. When we talk about cardiovascular disorders, we're really looking at issues like hypertension, coronary artery disease, heart failure, and arrhythmias. Each of these conditions can have a profound impact on a person's quality of life, and they often share common risk factors such as poor diet, lack of exercise, smoking, and genetic predisposition. Understanding these key points helps us see the bigger picture, how lifestyle choices influence cardiovascular health and the importance of early detection and management. Connecting this to our overall theme, it's clear that prevention and timely intervention are essential. Managing risk factors can significantly reduce the burden of these disorders. So, in essence, awareness and proactive measures are our best tools in combating cardiovascular diseases. Steroids are compounds that contain a specific type of ring structure known as a cyclic steroid nucleus, or what we call cyclopentanoperhydrophenanthren, often abbreviated as CPPP. Now, this ring system is quite interesting because it consists of a phenanthrene nucleus, which includes rings A, B, and C, and to that, a cyclopentane ring, which we refer to as ring D, is attached. This particular structure is fundamental to understanding how steroids are built and function in biological systems. The arrangement of these rings gives steroids their characteristic shape and properties. Additionally, when a steroid molecule contains one or more hydroxyl groups, that is, OH groups, it's typically called a sterol. These hydroxyl groups are important because they influence the molecule's solubility and biological activity. Understanding these structural features helps us see how steroids can serve various roles, from hormones to components of cell membranes. This overall structure and variation, such as the addition of hydroxyl groups, are key to their diverse functions in the body. There are several steroids present in the biological system, and they play crucial roles in various physiological processes. These include cholesterol, which is fundamental as a structural component of cell membranes and a precursor for other steroids. Bile acids are another group, essential for the digestion and absorption of fats. Vitamin D, on the other hand, is vital for calcium regulation and bone health. We also have sex hormones, such as estrogen and testosterone, that regulate reproductive functions and secondary sexual characteristics. Adrenocortical hormones, like cortisol and aldosterone, are involved in stress response, metabolism, and electrolyte balance. To give you some concrete examples, these steroids are interconnected, with many derived from cholesterol, highlighting its central role in steroid biosynthesis. Understanding these different steroids helps us appreciate how they contribute to maintaining overall health and homeostasis in the body. These are groups of bioactive PUFA, or polyunsaturated fatty acids, that serve as lipid messengers in the body. One key group of these messengers is the eicosanoids. Eicosanoids are generated through the oxygenation of 20-carbon polyunsaturated fatty acids, and the main precursor is arachidonic acid. Just a quick note on terminology. Icosa means 20 in Greek, which refers to the 20 carbons in these fatty acids. Eicosanoids act as local-acting hormones, meaning they influence nearby cells rather than traveling through the bloodstream to distant sites. This local action allows for precise regulation of various physiological processes, such as inflammation and blood flow. Because they work so quickly and locally, they have a very short half-life. They're usually released immediately after synthesis and act right away. Understanding eicosanoids is important because they highlight how lipid-derived molecules can coordinate complex cellular responses, fitting into the overall theme of how bioactive lipids regulate health and disease. Eicosanoids fall into two main classes. These classes are essential because they help us understand how these signaling molecules function in the body. Essentially, eicosanoids are lipid mediators derived from arachidonic acid, and they play key roles in inflammation, immunity, and other physiological processes. The two main classes are prostaglandins and leukotrienes. Prostaglandins are involved in processes like vasodilation, pain, and fever, which are all part of the inflammatory response. Leukotrienes, on the other hand, are primarily associated with allergic reactions and bronchoconstriction. Recognizing these two classes helps us appreciate the complexity of eicosanoid signaling and how they impact health and disease. This classification is a cornerstone in understanding how our body regulates inflammation and immune responses. They have ring structures, for example, prostaglandins, thromboxanes, and prostacyclins. These are all types of prostanoids, which are derived from arachidonic acid. Prostaglandins, in particular, play a crucial role in our body by modulating blood pressure, influencing uterine contractions during labor, and signaling inflammation. We often designate prostaglandins with letters like PGA, PGD, PGE, and PGF. These labels depend on the specific functional groups attached to the cyclopentane ring, which in turn determine their particular effects. Understanding these prostanoids helps us appreciate how the body finely tunes processes like blood flow, reproductive functions, and immune responses. It's fascinating how such small molecules can have such broad and vital impacts across different systems. Now, let's take a closer look at these molecules. They consist of leukotrienes, lipixins, and hydroxyl eicosapentaenoic acids, commonly known as HETEs. These are all derived from leukocytes, which are key players in our immune response. Structurally, they contain 3 carbon-carbon double bonds arranged in conjugation. This conjugated system is important because it influences their biological activity. Among these, the most significant leukotrienes in humans are LTA4 and LTB4. These molecules play crucial roles in inflammation and immune signaling. Additionally, we refer to these as linear eicosanoids because of their straight-chain structure, which distinguishes them from other eicosanoids that may have cyclic forms. Understanding these compounds helps us appreciate how our body regulates inflammation and responds to injury or infection. Let's take a look at this slide, which provides an example of eicosanoids. Eicosanoids are a fascinating group of signaling molecules derived from fatty acids, specifically arachidonic acid. They play crucial roles in various physiological processes, including inflammation, immune responses, and even regulation of blood flow. What's particularly interesting is how these molecules act locally near the site of their production, making them key mediators in cellular communication. As we see here, different types of eicosanoids, such as prostaglandins, leukotrienes, and thromboxanes, have distinct functions, but all originate from similar precursors. Recognizing these examples helps us understand how lipid mediators are integral to maintaining homeostasis and responding to injury or stress. This ties back to the overall theme of how bioactive lipids influence health and disease, highlighting their importance in both normal physiology and pathological conditions.